Okay, all set. Floor is yours, Weonjung.
Yes, good morning, everyone. I'm Weonjung Sohn, and today for my CA3, I'd like to talk about my research topic, which is advanced membrane bioreactor for sustainable urine valorization into a fertilizer in circular economy systems. So this is the contents of today's presentation, and I'd like to start from the background of my research. As we already know, the exponential growth of the global population has resulted in the significant increase in fertilizer use globally over the last 6 decades. And as the global fertilizer markets are expected to grow steadily in the next years, the Australian fertilizer markets are also expected to grow significantly by 2030. So we are currently living in a linear economy where we are heavily relying on the synthetic fertilizers. So we take the nutrient resources and make a synthetic fertilizer and use it, and they are removed in the wastewater treatment plants and then disposed to the water bodies. So in this linear economy, the production of synthetic fertilizers contributes around 2% of global energy consumption and also results in a large greenhouse gas emissions and depletion of limited resources. In addition, the excessive nutrients discharged into water bodies can pose a major global environmental challenge like eutrophication. So given this situation, our human urine is like liquid gold because it's rich in essential nutrients for plants, which are nitrogen, phosphorus, and potassium. Actually, urine only accounts for 1 volume percent in the whole municipal wastewater. However, it accounts for the 80% of nitrogen and 50% of phosphorus and 70% of potassium in the wastewater stream. So therefore, by source separating urine and transforming urine into a fertilizer, we can close this loop to make our circular economy. So by doing so, we can improve the conventional wastewater treatment plants to be more energy efficient and cost effective. And also it can potentially offset 13% of the global fertilizer consumption. If you have a look at the composition of urine, so one person produces average 1.2 liters of urine per day. And although the composition can be different depending on the diet or general health of individuals, fresh urine consists of this 95% of water and 2% of urea, nitrogen, and the rest of other ions. And the composition of fresh urine and stored urine can be different due to the urea hydrolysis. So in the fresh urine, most of nitrogen exists in the form of urea, and the pH is between 4.2 to 6.9. However, as soon as it contacts the toilet walls or pipes, urea is hydrolyzed into ammonium in very short time. And during this urea hydrolysis, because of alkalinity produced, the pH level increases up to 9.5. And most nitrogen is in ammonia nitrogen form. In addition, due to the high pH, phosphorus is likely to precipitate as a struvite. So therefore, direct use of stored urine is not recommended because of smell due to volatile ammonia and has unwanted micropollutants and organics. So many previous studies have investigated various technologies to convert urine into a more convenient fertilizer to use. And the technologies can be divided into physical, chemical, electrical, and biological methods. However, most technologies have limitations such as nitrogen loss, presence of micropollutants, or the recovery of only single nutrients, and most importantly, requirement of heating energy and chemicals. So therefore, nitrification, which means the biological ammonia oxidation, can potentially be the most promising technology, which can overcome these previously mentioned drawbacks. So urine nitrification, especially in this ultrafiltration membrane bioreactor with activated carbon, can be an innovative solution. In the bioreactor, ammonia oxidizing bacteria, AOB, converts ammonia to nitrite, and then the nitrite oxidizing bacteria, NOB, converts nitrite to nitrate. Therefore, the maximum 50% of ammonium to nitrate conversion and also the pH reduction without any chemicals addition can be achieved. At the same time, by using the ultrafiltration membrane, virus can be rejected and the high organic contents and micropollutants can be removed by the absorption on the activated carbon. So finally, nitrogen, phosphorus, and potassium can be recovered in one final product as an NPK liquid fertilizer. So as you might already know, in this building, this UTS building 11 has urine diversion systems which can separate urine from waterless male urinals in every floor. Because of this system, my research group is focusing on the urine collection from the building basement and urine processing in the MBR system, and finally the application of produced liquid fertilizer achieving nutrients in a circular economy. So both lab scale and pilot scale MBR have been studied for urine nitrification. And recently, our previous lab scale studies demonstrated that PAC addition to urine MBR system showed more than 99% of removal of micropollutants and also improvement in total organic carbon removal rate. So as a result, stable nitrification in a PAC-added UF MBR could produce ammonium nitrate solution, which can be safe, effective liquid fertilizer. So moreover, our urine fertilizer produced from our MBR system has been applied to various hydroponic systems under different conditions to grow parsley, ripsalis, coriander, and basil in the last few years. And these results from these case studies found that our urine fertilizer was comparably effective to commercial fertilizer in the hydroponic systems. This figure on the left-hand side shows the schematic diagram of urine MBR system. So traditionally, pH dosing pump has been used for the pH-based urine feeding to MBR. So whenever the pH level decreases due to the alkalinity consumption during the nitrification process, the dosing pump automatically doses the high pH urine to maintain the reactor pH at 6.2. So as a result, the hydraulic retention time, HRT, was regulated based on the urine dosing rate, which depends on the nitrification rate. So the remaining challenge of this urine MBR system can be a long hydraulic retention time, HRT, which was reported to be 10 to 14 days in the previous studies. So this long HRT makes the system large footprint and more energy consumption, and ultimately can be a barrier for the commercialization opportunities. So therefore, my research is focusing on overcoming this challenge, how to improve the nitrification efficiency, thereby reduce the HRT of the MBR system. As such, my thesis has 4 research objectives corresponding to each technical chapter. Firstly, in chapter 3, I focused on the investigating the effect of temperature and dissolved oxygen, DO conditions in MBR treating urine on the nitrification performance and the resulting HRT to identify the optimal temperature and DO conditions. And in chapter 4, I focus on exploring the performance improvement of a biofilm carrier supported MBR with the PAC in comparison to the conventional MBR in terms of nitrification and HRT, as well as the microbial community composition. The following chapter evaluated the feasibility of ammonia air stripping as a pre-treatment method for reducing the ammonia concentration in the hydrolyzed urine to enhance the stability and the efficiency of nitrification in the MBR. And the last chapter here focused on investigating the effect of fixed HRT conditions in a pilot scale compact MBR system on the urine nitrification performance and examine the optimal HRT threshold by assessing the efficiency of the produced liquid fertilizer on the hydroponic plant growth. So as a first step to reduce the HRT, I investigated the effect of temperature and dissolved oxygen conditions in urine MBR. So temperature and dissolved oxygen, DO conditions, are very important parameters for nitrification. So previously it was reported that at the temperature below 16 degrees, NOB growth rate was higher than AOB. However, the above 16 degrees, AOB growth rate was exponentially increases. Regarding the DO levels, the growth rate can be declined below the DO level of 3 to 4 milligram per liter, while it can be increased up to 5 milligram per liter of DO. Although they are very important operational factors, our urine MBR studies have only been conducted at the room temperature at the constant DO. So therefore, this study aimed to investigate the optimal operational conditions for urine MBR performance, focusing on the nitrification and HRT, and specifically the effects of three different temperature conditions, which are 10, 20, and 30 degree, and combined with the 3 DO levels, 2, 4, and 6 milligram per liter, resulting in 9 operational scenarios were evaluated. For the experiment, so 3 double-jacketed reactors with effective volume of 4 liters were set up, and each reactor was set at 3 different DO levels. So 2 milligram per liter of DO for reactor 1, and 4 milligram per liter for reactor 2, and 6 milligram per liter for reactor 3. So as can be seen in the temperature variation graph, the reactors were sequentially adjusted to operate at 10 degrees and 20 degrees and 30 degrees by effectively controlling the temperature using this circulator. And each temperature condition was maintained for 30 days with the 10 days of transitional period between the temperature adjustments. The reactors were operated at the pH-based feeding mode using the pH dosing pump. Figure A, B, and C are showing the nitrification results in each reactor over the whole operational period. So key findings from here were that the temperature increase from 10 degrees to 20 degrees enhanced the nitrification and reduced the HRT across all reactors, while the reactor 2 with 4 milligram per liter of DO achieved the optimal performance. And further increase to 30 degrees resulted in nitrite accumulation in all reactors, while in reactor 3 with 6 milligram per liter of DO had the highest nitrite accumulation up to 130 milligram per liter due to the increased AOB growth rate than NOB. Also in reactor 3, under the highest DO level, the nitrite accumulation observed in all temperature conditions, and this might be due to the higher oxygen affinity of AOB that favor the AOB growth and metabolism. And in terms of the total organic carbon removal rates, all reactors showed more than 80% of TOC removal rates across all temperature and DO conditions. And it showed the highest at the 20 degrees regardless of DO levels. However, in reactor 1 consistently showed the lowest TOC removal rates in every temperature conditions, indicating that this low DO level can be less effective. And the highest TOC removal rate of 89% was achieved at 20 degrees with 4 milligram per liter of DO, highlighting that the synergistic impact of moderate temperature and DO levels in the reactor performance. To investigate the effect on the biomass growth, MLSS, which is mixed liquor suspended solids, and MLVSS, mixed liquor volatile suspended solids, were monitored, and also the sludge volume index were measured. The MLSS concentrations were uniformly adjusted to 5.8 gram per liter before starting each temperature conditions. And at the 10 degrees in reactor 1, the MLSS increased the most. However, the MLVSS to MLSS ratio reduced, as can be seen here. And this indicates more growth of non-volatile solids. And in reactor 3, minimal growth of MLSS and MLVSS was observed. and also constantly high SVI values were observed, and which shows the reduced sludge settlability, indicating the poor sludge flow formation. On the other hand, in reactor 2, the MLSS and MLVSS had a steady growth, and the MLVSS to MLSS ratio was relatively stable at around 0.8. So in all reactors, the temperature increase to 30 degrees showed high SVI values, indicating the reduced sludge settlability, because the high temperature affected the functioning of these extracellular polymers, leading to the low bioflocculation abilities. And this table is showing the microbial diversity results in each conditions. So ACE and CHOW1 indicates species richness, while the Shannon and Simpson measures the species evenness. So regarding the impact of temperature, key finding was that this 20 degrees temperature condition resulted in the highest microbial evenness and richness compared to 10 degrees and 30 degrees. This might be because any vulnerable or temperature-sensitive microbial species reduced at the extreme temperature conditions, leading to the decreased diversity. In terms of impact of DO, among the 20 degrees conditions, this 4 milligram per liter of DO resulted in the lowest diversity, indicating the selective enrichment of nitrifying or organic matter removing bacteria, because the dominance of certain species under preferred conditions contributed to the reduced overall microbial diversity. Moreover, the functional bacterial communities analyzed at phylum and genus levels, with detection of 10 major phyla and 37 major genera. So the top 4 major phyla were consistent in all conditions, while the proteobacteria dominated in all samples, and its relative abundance remained stable between 10 degrees and 20 degrees. But it increased up to 1.8 times at 30 degrees in all reactors. At 30 degrees, the 2 milligram per liter and 4 milligram per liter of DO, which is R1 and R2, dominated by beta proteobacteria class, while in reactor 3, it was dominated by gamma proteobacteria. So overall, although there were some shifts in microbial compositions under different temperature and DO conditions, the dominance of major phyla and genera across all the reactors reflects their excellent performance in organic matter removal. This figure is showing the relative abundance of only nitrifying bacteria, AOB and NOB. So the predominant AOB was found to be the Nitrosococcus, and predominant NOB was Nitrospira. So as indicated in green color, at 10 degrees, AOB Nitrosomonas was likely to show an increased relative abundance compared to other temperature conditions. However, the temperature increase to 30 degrees showed remarkable increase of AOB of up to like 3 times, especially the Nitrosococcus. In addition, at the 10 degrees and 20 degrees in R1 and R2, Nitrospira, colored in purple, was more abundant than AOB, reflecting the stable nitrification. However, in R3, with the highest DO, showed the greater AOB abundance at all temperature conditions compared to R1 and R2. These results are reflecting the persistent nitrite accumulation events. In conclusion of this chapter, so the optimal nitrification performance was observed at the moderate temperature and DO levels, which is 20 degrees and 4 milligram per liter, supporting the effective organic matter removal and also biomass growth. And microbial dynamics were significantly influenced by temperature and DO conditions, with the composition and abundance of AOB and NOB showing the variable responses across different conditions. And lastly, the high temperature and high DO led to the increased AOB abundance, resulting in the nitrite accumulation, highlighting the importance of maintaining the balanced operational conditions for effective MBR performance. So in Chapter 4, the MBR was incorporated with biofilm carriers and powdered activated carbon for the enhanced biological nitrification of urine. So as another approach to address the remaining challenge of long HRT, biofilm carrier addition was investigated in this study. So carriers addition can support more biomass, especially for slow-growing nitrifying bacteria, by supporting their attached growth. So leading to the nitrification enhancement and the HRT reduction for the compact and high-performance MBR system. Therefore, the research objectives of this chapter was firstly investigating the effect of biofilm carriers and PAC incorporation in overall performance, including the nitrification rate and HRT, compared to the conventional MBR. And after that, the microbial diversity and composition was investigated. And finally, the effect of incorporation on the nitrifying bacteria was also explored. So this figure is a schematic diagram is incorporated MBR, and the biofilm carrier used in this study is a commercially available carrier having high surface area, and its parabolic shape and low diffusion depth can have high oxygen and nutrient diffusion for the active biofilm formation, and also it can prevent the anoxic zone for the denitrification. So this biofilm carrier was added from the beginning of the operation to support the bacterial growth. However, the PAC was added after this carrier's surface saturation and started to sink down to avoid PAC accumulation on the carrier. Figure A and B here are showing the nitrification results from the conventional MBR without any additives and the biofilm carrier and PAC incorporated MBR. And as a result, the incorporation of carriers and PAC resulted in the shortened startup time by 14 days compared to the conventional MBR. And as colored in black, compared to the conventional MBR, the incorporated MBR showed 26% faster recovery time from this accumulation event. And during the second phase of each MBR, the nitrification rate was higher in the incorporated MBR by 36%, thereby the HRT could be reduced by 40% from 10 days to 6 days. So in terms of the suspended biomass growth, which is MLSS and MLVSS, the conventional MBR showed the steady increase with an increase with in the MLVSS to MLSS ratio up to 0.88. However, in the incorporated MBR, the MLSS growth was more obvious after the PAC addition here, with an increase of the ratio up to 0.9. Regarding the attached biomass on the carriers, during the startup phase, it increased to 2.8 gram, indicating the effective biofilm formation. However, in the second phase, it regulated this attached biomass was maintained at 3 gram of total mass, which means the biofilm layer thickness was well maintained without any accumulation. These figures are showing the total organic carbon removal rates of each reactor. In the conventional MBR, the removal rate was maintained at around 85%. In the incorporated MBR, the 2 gram per liter of PAC was added in the second phase here, and the removal rate improved by 11% up to 96%. And this can be because of the organic absorption capacity of PAC. As a result of microbial analysis, this table 2 is showing the sequencing depth and observed operational taxonomic units, OTUs, along with the diversity estimator as a Shannon index for each sample. And rarefaction curves were also generated based on the observed OTUs to examine the difference in microbial richness. The diversity and richness were highest in the seed sludge and then reduced by transition feed water to source-separated urine in the conventional MBR. However, in the MBR with the incorporation, even lower diversity and richness was observed. And this result might be because the addition of pH and biofilm carriers was able to create a selective force that enriched certain species, making them predominant microbes in the system. And further taxonomic analysis down to phyla and genus level was conducted for more thorough and detailed information on the bacterial communities. As a result, the feed water changed to source-separated urine from the seed sludge significantly shifted the dominant sequence order from the seed sludge. And interestingly, PAC addition did not really change the order of dominance among the microbial phyla. However, it resulted in an increase in their abundance by up to 3%. So overall, the incorporated MBR highly enriched the proteobacteria and firmicutes, which explains the better TOC removal rates in the incorporated MBR. After that, we had a closer look at the enriched nitrifying bacteria in different growing form. So the acclimation of seed sludge with the source-separated urine highly enriched the predominant AOB, Nitrosococcaceae, and NOB, Nitrospira, in the suspended sludge by up to 30-fold and 20-fold, respectively. And the predominant AOB, Nitrosococcaceae, showed 7-fold higher enrichment by attached growth on the biofilm carriers rather than the suspended form. So overall, comparing the abundance of nitrifying bacteria between the two reactors, the incorporated MBR significantly enriched the abundance of nitrifying bacteria compared to the conventional MBR. So in conclusion, biofilm carrier and PACA incorporation highly improved the nitrification rate and following HRT and the TOC removal rate as well. And a comprehensive analysis of microbial dynamics in the incorporated MBR demonstrated the abundance and distribution of key functional genera, especially the nitrifying bacteria. And finally, this enhancement suggests the feasibility of more compact and highly efficient MBR system for source-separated urine nitrification. So in Chapter 5, as another approach to reduce the system HRT, the ammonia air stripping of urine was investigated as a pretreatment for MBR nitrification by reducing the ammonia concentration in the feed urine. Many previous studies have focused on the ammonia stripping of urine to recover nitrogen, and they were focused on air stripping and thermal or vacuum thermal stripping, which requires additional chemicals for alkalization or energy inputs for heating to improve the efficiency. However, these significant energy inputs and additional chemical limit their economic feasibility. Hydrolyzed urine, as it is, has a high pH of 9.5, which can be sufficient to strip 50 to 60% of ammonia at the room temperature, reducing the ammonia concentration in the MBR feed to below 2 grams per liter, thereby improving the MBR nitrification rate. So in this study, I aimed to explore the impact of ammonia air stripping on the hydrolyzed urine, utilizing its natural high pH for efficient ammonia removal at the room temperature. The feasibility of recovering ammonia as a potential ammonium sulfate fertilizer was also assessed by optimizing the ammonia capture efficiency using a 2-stage sulfuric acid solution following the air stripping process. The ammonia stripped urine was then fed to the MBR system to evaluate the system stability and nitrification efficiency, thereby assessing the effect on the system's overall HRT. This figure here is showing the experimental design. So the raw hydrolyzed urine was aerated at the room temperature without any chemical pH adjustments. And the stripped free ammonia and air transferred to the 2-stage sulfuric acid solutions to absorb ammonia, and potentially this can be recovered as ammonium sulfate fertilizer. And the exhausted air here after the ammonia absorption process can be potentially reused for the NBR aeration to improve the energy efficiency. Then the ammonia stripped urine became the feed to the NBR to investigate the nitrification. So firstly, in the ammonia stripping process, air flow rate is one of the most important operational factors which affects the ammonia stripping efficiency, as it directly impacts the mass transfer rate between the liquid and gas phases. So firstly, to optimize the air flow rate, the 5 different flow rates, 0.5, 1, 2, and 2.5, and 3 liter per minute of air flow rates were tested and measured the ammonia removal rates over time. So as a result, higher flow rates resulted in better ammonia removal efficiency. And as can be seen in this table, the 68% of ammonia removal efficiency in 2-day aeration achieved by the 3 liter per minute of air flow rate. However, further increase in the air flow rate was not feasible due to the excessive urine foaming. After that, to see the ammonia recovery by absorption, 2 stages of 0.5 more of sulfuric acid solutions were sequentially connected to the aeration tank for 48 hours with the flow rate at the 3 liter per minute. So in this first trial, the acid solution bottles had 500 milliliters of volume for each bottles. And as a result, only 31% of stripped ammonia could be recovered. The reason might be due to the low mass transfer rate from insufficient volume and height of these acid bottles. So therefore, in the second trial, to increase the mass transfer rate, the higher height of the acid bottle, but in bottle 1 and 2 was investigated. So the result showed significantly increased recovery rate to 51%. And these findings suggest that further optimizing the absorption stages, specifically by increasing the mass transfer rate and adjusting the concentration of sulfuric acid, can significantly enhance the ammonia recovery rate, enabling the effective capture of ammonia as ammonium sulfate fertilizer. Followed by the stripping process, lab-scale NBR was started to operation from the sludge acclimation, since the ammonia stripped urine has higher COD to N ratio than the raw hydrolyzed urine. So the NBR was started up for 35 days for acclimation of sludge by feeding diluted 48-hour stripped urine with an initial nitrogen concentration of 200 milligram per liter. So throughout the startup phase, no nitrite accumulation occurred, and the conversion rate of ammonia to nitrate ranged between 50% to 70%. And the total organic carbon removal rate remained at 87% of excellent removal efficiency. So this NBR experiment is still ongoing, and in the subsequent phase, 48-hour stripped urine will be fed without any dilution, and followed by the introduction of 24-hour stripped urine to examine the HRT. So in conclusion, the air stripping of hydrolyzed urine conducted at the room temperature demonstrated that the highest removal efficiency at an airflow rate of 3 liter per minute, achieving the ammonia removal rate of 61% and 68% after 24 hours and 48 hours of aeration, respectively. And ammonia absorption into sulfuric acid solution reveals that the significant increase in recovery efficiency by enhancing the mass transfer rate, highlighting the potential feasibility of ammonia recovery following the adequate optimization of the process. And the subsequent biological nitrification of ammonia-stripped urine in the MBR indicated that this successful sludge acclimation during the startup time. So unfortunately, the experiment for this chapter is still ongoing. However, it will be finalized within the next month and will be included in the thesis. So in the last technical chapter, the urine MBR has been scaled up to pilot scale compact system to investigate the impact of HRT conditions and hydroponic applications. So based on our lab scale studies, we developed this pilot scale compact MBR system. And in this study, I focused on investigating the effects of different HRT conditions on the nitrification and the following product fertilizer effectiveness for the application in the hydroponics. So since the previous urine treating MBRs were typically operated at the pH-based feeding mode to achieve a stable nitrification. So the HRT was a dependent factor of nitrification rate. However, as I mentioned earlier, this extended HRT in the MBR system was the main challenge and a barrier for the potential commercialization of the system. So this study aimed to investigate the impact of fixed HRT under continuous feeding mode on the urine nitrification performance, and the produced effluents from different HRT conditions having different nitrogen compositions were applied to the hydroponic basil and orchard grass growth to see the optimal HRT threshold. So the pilot scale compact MBR system, as can be seen in these photos here, has a main display for controllers and an inside controller panel. And this also has maximum 200 liters of MBR tank inside, together with this 100 liters of effluent tank. This figure is showing the schematic diagram of compact pilot scale MBR system. So the hydrolyzed urine from the feed tank is pumped into the MBR tank using an internal influent pump. And the effluent pump, which is connected to the UF membrane modules within this MBR tank, produces the permeate, which is then stored in the permeate or fertilizer tank here. So this fertilizer can be directly applied to the parklands or gardens after simple dilution. So this compact MBR system is therefore showing the feasibility of on-site urine treatment and fertilizer production system in a building or household scale, and potentially application to the urban agriculture systems. This figure is showing the nitrification performance in the MBR system over the period of 160 days. So during the startup time, diluted urine was fed to the system for the acclimation of sludge bacteria. And in the phase 1, undiluted urine started to be used as a feed under the pH-based feeding mode as typical. And the average HRT resulted in 7 days. Then from phase 2 to 4, the urine was fed continuously by intentionally operating at a reduced fixed HRT conditions, which are 5 days, 3 days, and 1 day. So the result showed significantly increased nitrite concentration from these 3-day HRT conditions, and further increase to 1-day HRT condition showed a lot of nitrite accumulation here. And this systematic HRT reduction in each phase also showed this gradual decrease in ammonia to nitrate conversion rate from 40 to 30 and 20 and 10% in each phase. We also conducted the microbial analysis of sludge samples from different HRT conditions, and the result showed the microbial diversity was reduced when the HRT reduced to 1 day, which is phase 4. And the microbial composition and phylum level and genus level showed the order of dominant phyla and genera have been significantly changed from phase 3. However, the abundance of proteobacteria increased with the HRT reduction. So overall, the Mycobacterium and Firmicutes emerged as a predominant genera in all samples, contributing to the excellent TOC removals. This heat map is showing the relative abundance of AOB and NOB genera in each phase. So the dominant AOB was Nitrosococcus, and the dominant ANOB was Nitrospira. In phase 1, combined AOB relative abundance was around 1.4%, and the NOB was highly abundant at around 7%, resulting in the stable nitrification. In phase 2, increased the feeding rate at the HRT 5 days resulted in higher AOB abundance as well as an increase in NOB abundance as well to 8%. However, further increase in feeding rate to 3 days of HRT caused a major shift in AOB and NOB populations, notably enriching the Nitrosococcus AOB to 13% and 14% with the HRT 3 days and 1 day respectively, while the Nitrospira NOB was significantly inhibited, contributing to the significant nitrite accumulation in the system. So after that, the permeate from each phase has been collected for hydroponic test on basil and grass growth in the commercial indoor hydroponic system called hydro gardens. And they were cultivated for 12 weeks for the basil and 6 weeks for the grass. So in this table, we could see the final diluted treatment solutions for hydro gardens have different nitrogen compositions, meaning the ratio of ammonium, nitrite, and nitrate were different in each solution. However, the total nitrogen concentration was maintained at 250 ppm across the hydro gardens for the comparison of the growth. So the basil and grass grown with the different treatment solutions from each phase has been quantified in terms of the number of stems, height of main stems, as well as the fresh biomass yield and dry biomass, and also the roots to shoots ratio. So as a result, the number of stems of basil showed gradual decrease along with this HRT reduction, while there was no significant variances in the height of main stems among the hydro gardens of basil. And the stem numbers of grass had slight decrease from the HRT 1 day. However, the height of main stems of grass stayed at the similar level across the hydro gardens. The biomass yield of basil, which is a fresh weight of this shoot, significantly decreased from HRT 3 days, while the grass biomass yield showed this similar yield among the hydro gardens. Most importantly, in terms of this roots to shoots ratio, HRT 7 days and 5 days showed comparable values to the commercial fertilizer in both basil and grass, meaning the balanced growth of roots to shoots. However, the ratio was notably higher in HRT 3 days and 1 day, which might indicate more nutrient allocation to the roots as a response to the stress conditions. So overall, the urine fertilizer produced from up to HRT 5 days showed the comparable results to the one grown with the commercial fertilizer, which can be an effective optimal HRT condition. However, the reduced HRTs at around 3 days and 1 day showed inhibited growth of basil and grass, which means they might need additional treatment to reduce the high nitrite concentration. However, interestingly, the effect of this inhibition was more obvious in basil growth compared to the grass growth. So in conclusion, this study was conducted to evaluate the effects of different HRT conditions on the nitrification performance and fertilizer effectiveness to support the potential commercialization of this compact MBR system. And the system was acclimated well to a 7-day HRT under pH-based feeding mode. However, as the HRT was gradually reduced, at the fixed HRT condition, the ammonia to nitrite conversion rate was decreased gradually in each phase, accompanied by the increasing nitrite accumulation. The hydroponic application of urine fertilizer demonstrated that the HRTs of up to 5 days provided optimal basil growth comparable to the commercial fertilizers, while the grass was less affected by the HRT conditions compared to the basil. So in the last chapter, this chapter is a conclusions and recommendations. So in conclusion, my thesis aimed to improve the urine nitrification rate, thereby reducing the HRT, which is one of the primary challenges in urine treating MBR systems. So to address this, 4 approaches were investigated in the lab scale and pilot scale MBR systems. And in chapter 3, temperature and DO conditions were optimized, and the major results were that the 4 milligram per liter of DO under 20 degrees temperature showed the optimal nitrification rate and the shortest HRT. And chapter 4 focused on the integration of biofilm carriers and PAC, which improved the nitrification rate by 36%, thereby reduced the HRT by 40%. In chapter 5, ammonia air stripping of hydrolyzed urine showed promising potential as a pre-treatment for enhancing the nitrification in urine MBR systems. And in chapter 6, using the pilot scale MBR, the study investigated the transition from the traditional pH-based urine feeding to continuous feeding at the fixed HRT, which resulted in high nitrite levels in the hydroponic solutions while showing the potential of orchard grass as a target crop for the fertilizers produced under the less optimal conditions. Based on the results from this study, I would like to propose some recommendations for the future study. So firstly, while chapter 3 focused on the DO conditions, the aeration intensity and its impact requires further evaluation. So further studies can explore the various the air diffuser types or advanced technologies like micro or nano bubble systems. So based on the results from chapter 4, the biofilm carriers can be further explored in different configurations or designs, or commercially available bacteria-enriched carriers can also be tested. So although this study applied the PAC addition on both lab scale and pilot scale MBRs, the concentration or replenishment rate, also the long-term effects of PAC should be considered. Based on the chapter 5 study, the citric acid or acetic acid can be a good substitute for the sulfuric acid, considering its safety and handling issue. So another big challenge in the urine diversion system can be a struvite formation in the urine storage tanks and pipelines. So this can be redissolved in the ammonium sulfate solution, resulting from the stripping process, so that the complete nutrient recovery at a building scale can be feasible. And the urine fertilizer still needs to be applied in various plant types to assess its efficiency, and also optimization is also needed. And for the concentration of urine fertilizer, so although we have been used just a simple distillation process for the concentration, the energy efficient concentration technologies like electrodialysis can be explored further. And lastly, further scaling up to full scale and also long-term monitoring will be the remaining challenge for the future study. I'd like to express my gratitude to my supervisors, Professor Sohn and Dr. Sharap, and also my group members. Thank you for listening my CA3 presentation, and sorry for the delay time. Thank you.
Okay, thank you, Weonjung, for your good talk.